Hi everyone, my name is Gabriela Jessica and today I'm going to talk about my research paper titled The Comparative Analysis of NATM Gene Function in Simple Leaf of Arabidopsis thaliana and Compound Leaf of Cardamine hirsuta. So let's start from the leaf. The leaf itself is an essential part of the plant, serving as a site for vital processes such as photosynthesis. And across plant species, there are diverse leaf shapes ranging from simple leaf to a more complex leaf or compound leaf. Simple as in has a single blade and continuous lamina, and compound that has a subunit called leaflet that appeared like a single leaf. And in each plant there are also a variety of leaf margin. It can be smooth, it can be serrated, or it can be looped. And this variety of leaf morphology significantly affect productivity and survival. For instance, it was found that the genetic modification of leaf angle in rice improved light interception in the canopy and resulted in higher grain yield. Now, the leaf development is regulated by transcription factors such as tail homeodomain proteins or 3-amino acid loop extension. And tail homeodomain proteins are conserved in plants and consist of two classes. The first one is knotted one-like homeobox or NOX, and the second one is PAL1-like. NOX protein consists of three classes, class 1, class 2, and class 3, and NATM falls into the third class of the NOX gene family. The characteristic is that NATM encodes the main NOX domain that NOX protein have, but it lacks the homeodomain. But it still can form dimers with other NOX proteins and BAM proteins. In previous research, it was found that the overexpression and ectopic expression of NATM in Arabidopsis thaliana caused increased leaf serration. However, the NATM loss of function phenotype is not yet known. Interestingly, members of PAL genes, SO1 and SO2, also affect leaf shape. The double mutation of SO1 SO2 in Arabidopsis thaliana caused increased leaf serration. So the overexpression of NATM and the phenotype of SO1 SO2 mutation in Arabidopsis thaliana resemble each other, which show an opposite effect. In this project, the role of NATM in different species and leaf morphology will be identified to elucidate the possible species-specific regulatory mechanism of NATM. And in this project, two plants will be used. The first one is Arabidopsis thaliana, which is a model plant that have simple leaf, and the second one is Cardamine hirsuta, which is closely related to Arabidopsis thaliana but have compound leaf. These two plants are useful because they are related to each other but have different leaves. So they are useful for comparing the role of NATM in different leaf shape. The aim of this project is to examine loss of function NATM mutation in Arabidopsis thaliana and generate CRISPR-induced null mutation in Cardamine hirsuta using CRISPR-Cas9 editing, and lastly to observe and compare the function of NATM in both plant species. The hypothesis of this project is that the inactivation of NATM in Arabidopsis thaliana will cause the reduction of leaf serration, while the inactivation of NATM in Cardamine hirsuta will cause reduced compound leaf complexity. And here I am going to explain the two most critical methods. So after the design of CRISPR construct, transformation, and seed screening, the next step is to screen the CRISPR-induced mutation. The method is using RFLP. So the first step is PCR using suitable primers, followed by digestion with restriction enzyme. For wild type, the restriction enzyme recognition site will still be intact, and therefore the enzyme will cut at the recognition site. However, CRISPR will alter the recognition site, and in mutant plants, the recognition site will not be there anymore, and the enzyme won't cut. And we can see this in the gel agrophoresis visualization with different band patterns. And the identified mutant plants were sent for sequencing. The confirmed mutant plants were then phenotyped, and for Arabidopsis thaliana phenotyping, the 8 rosette leaf were used to collect number of serration, serration depth, serration tip angle, and serration sinus angle. And statistical analysis was done to this data using the two sample t-tests for pairwise comparison. And the statistical analysis was done using Origin software. Now for the result and discussion of Arabidopsis thaliana. 3 different lines were screened for NATM mutants, and homozygous NATM mutants were found in wild-type backgrounds and in so-and-so-2 double mutant backgrounds. And these 2 lines were sent for sequencing. As a result, you can see here the type of mutation. The bold sequence here indicate the wild-type sequence, while the sequence underneath indicate the mutant sequence. And the pink square here is the type of mutation. For one line, it has 1 base pair insertion here, the other line has 11 base pair deletion, and 1 base pair change. And this mutation causes a frameshift mutation, which leads to a predicted non-functional NATM gene. These 2 lines were then phenotyped, and the NATM with wild-type background was compared to the wild-type lines, while the NATM with so-and-so-2 double mutation were compared to the so-and-so-2 double mutant. And it appears that the NATM mutation caused less and less noticeable suration. And to confirm this, statistical analysis were done. As a result, NATM mutation caused lower suration number, and it shows significant difference, while it did not show any significant difference for suration then. Now for the suration tip angle and suration sinus angle, the NATM mutation caused larger angles, and it all shows significant difference except for the so-and-so-2 and so-and-so-2 NATM for suration tip angle. So overall, NATM mutation caused fewer suration, larger suration tip angle, and larger suration sinus angle. So, NATM mutation caused fewer suration than wild type, which align with the hypothesis. And as we know from the introduction, NATM and so-and-so-2 has an opposite effect or antagonistic effect. And the result also indicate that NATM mutation modulates the improved suration caused by the so-and-so-2 mutation. Next, the leaf suration itself is regulated by auxin, PIN1, and COX2. So COX2 directs PIN1 convergence, which cause auxin maxima to be formed and also cause suration. And in previous research, it was found that so-and-so-2 mutation, although it did not change any expression patterns, it increased the auxin maxima and PIN1 convergence point, which caused increased leaf suration. And NATM are known to titrate so-and-so-2 and sequester BAL and NOX protein. So the NATM mutation can possibly lead to fewer auxin maxima and PIN1 convergence point, which can explain the fewer suration and larger angles. Now for the result and discussion of cardiomyine hirsuta. So 3 heterozygous TH-NATM mutant plants were identified and sent for sequencing. And you can see here the type of mutation that the 3 lines have. Same as before, the top bold sequence is the wild type, while the sequence underneath is the mutant sequence. And the red box here is the type of mutation. So the first line has 2 base pair deletion and 1 base pair change. The second line has 1 base pair insertion and 1 base pair change. And the third line has a 5 base pair deletion. And this mutation causes a frameshift mutation, which can lead to a predicted non-functional CH-NATM gene. So out of 3 lines, 2 were planted to obtain the homozygous CH-NATM plants. And preliminary phenotype analysis showed that some plants have leaflet fusion, as you can see here in the picture. So for NATM influence in compound leaf species, the hypothesis is loss of function of CH-NATM will cause reduced leaf complexity. And this project successfully generated the CH-NATM mutation, and preliminary observation showed a phenotype, but it still needs further analysis to confirm whether the phenotype is caused by NATM mutation or not. The possible mechanism behind this is that NATM is known to affect BAL and NOX complexes homeostasis, and this results in the promotion of NOX1 expression. And NOX1 is crucial for leaflet formation. So when CH-NATM is mutated, it could lead to reduction of NOX1 expression and cause reduced leaf complexity. So in conclusion, the NATM mutation in Arabidopsis thaliana caused fewer suration and also altered suration angles, while the NATM mutation in Cardomyine hirsuta potentially reduced leaf complexity and caused leaflet fusion. This can help explain how we can get different leaf shape in different species. And for future research, the research should focus on NATM's molecular pathway, interaction with other tails proteins, and other components such as auxin. The future research can also create the CH NATM mutation in SO1 and SO2 double mutant background to compare the NATM and SO1 SO2 interaction in both Arabidopsis thaliana and Cardomyini hirsuta. These are the reference used. Thank you for your attention.